afternoon and welcome to all. This is Ankana, the assigned moderator from IJCP Group. I wholeheartedly welcome all the delegates across the country. We are fortunate enough to get supported by the doctors. Thank you all the doctors for taking out time and join us today. I'm taking this opportunity to welcome today's master doctor, none other than the eminent speaker, Dr. Narsingh Verma, sir. Sir is the Secretary General at the Department of Physiology at King George Medical University, Lucknow. Now, with the help of Sir and his wonderful insights, we'll be taking a close look on today's topic. Join us for an in depth webinar on gastrointestinal physiology and the gut microbe, where we explore the intricate relationship between the digestive system and gut microbiota. The session will delve into the physiology of the gastrointestinal tract, examining how it functions and interacts with the diverse microbial community residing within it. Now, any further delay, sir, I'd like to hand over the session to you. Kindly proceed with your talk. Over to you.
Thank you for giving this opportunity and talking about the GI physiology and gut microbe. How the gut microbes are essential for the normal functioning of the GI physiology. And here I will start my deliberation with a very short story. One day, body parts were fighting with each other as to who is the most important part of the body, as usually occurs in a team. Sometimes it occurs that who is the best out of the team. And we know that our body is made of trillion of cells, and out of these trillion of cells, the the cells make the particular organ, and organs make the organs unite and make the system, and systems unite to form a body. But the story is that the body parts are fighting that who is the best. The brain said, I am the most important and I control everything. The heart said, I am the most important as my beating keeps the body going. And liver said, no, I am the most important as I help in metabolism, excretion, and detoxification, and so many other works. The liver said, and after that, the gut said, I am the most important as I digest, absorb, protect, excrete, produce hormone, reabsorb, so many things. So, gut finally said that I am the best. Everyone laughed at the gut, but the gut got annoyed and angry and shut down. Within a few days, every other body system agreed that gut was the most important. So, gut is the most important part of the body. Our ancestors were knowing that. Somehow, we have forgotten, and we are burdening our gut daily, day by day. So, all diseases begin in the gut. That was said by Hippocrates around 460 BC. And health is determined by the microbiota of our gut. Again, Hippocrates. Most treatable disease arise from the presence of ama, a toxic byproduct of poor digestion. So basically, ama or amla, they said ama, but they thought in Ayurveda that every disease is because of changes in digestion. And they are talking about treatable disease. And we are also interested to know about treatable disease. About untreatable disease, we are not interested because we can't do anything. So, if we are able to do anything, that is the diseases which are born out of the toxic substances which are byproduct of poor digestion. 70% of our immunity system is located in our gut. So again, immune system, here again the gut is important. So most of our immunity arise out of the gut. Gut microbiota, I will talk briefly about gut microbiota. When a baby is born, hardly any bacteria is present in the gut, but this notion is being challenged by the scientists. They say that the baby born depends upon whether it is a vaginal delivery or a cesarean section. The microbiota will depend upon this, whether it is a vaginal delivery or a cesarean section. Today, I will not talk much about this because this is not my topic today. So, gut microbiota, they are not present or very less in amount when the baby is born, but after that, the gut starts developing a lot of microbiota, and the whole ecosystem of the gut microbiota is called as biome or microbiome. So each one of us consists of approximately about 40 trillion human cells and 22,000 human genes. Trillion cells, 40 trillion, and about 22,000 human genes. And human gut hosts 100 trillion microbes, more than our cells. They are called as microbiota. And 10 million microbial genes, 22,000 human genes, and 22,000 human genes, and 10 million microbiota genes, which are called as metagenomes. So our genome is lesser than metagenome. So metagenomes become very, very important, weighing an average of around 1.5 kg. About 1.5 kg bacteria are present in our gut. Microbiota changes from infancy to adulthood. In infancy, in beginning, they are very less. They gain up. During old age, again, their constitution changes, number changes, and our intestine has less number of microbiota than the younger days. Bacteria from three major groups represent 95% of the total microbiota. The microbiota which are present in our gut, 95% of the total microbiota belongs to only three groups. There are other groups as well, but they are very less in number, only 5%. And these 95% of the total microbiota are either firmicutes, or bacteroidetes, or actinobacteria. Largest interface. Gut is the largest interface. The whole gut is the largest interface, 250 to 400 meter square. See how big it is. 250 to 400 meter square between the host and environmental factors. This is the interface and antigens in the human body. So it is the interface which determines how much antigens will go in and how much environmental factors are going to affect the host. In an average lifetime, around 60 tons of food pass through the human GI tract. 60 ton in average life I'm talking about. Through GI tract, 60 ton of food passes, along with an abundance of microorganisms, and these microorganisms come from the food particles or food which we take. And from environment, these bacteria or microbes go in, which impose a huge threat on the gut integrity. So if this is not managed properly, the gut integrity will be in threat. This I have already mentioned, around 100 times more genomics content than the human cells. The human gastrointestinal tract harbors a complex and dynamic population of microorganisms the gut microbiota, which exert a marked influence on the host during the homeostasis and disease. In both the condition, when a person is healthy and a person is diseased, in both the condition, microbiota, gut flora, they influence us very much. Multiple factors are contributing to the establishment of a human gut microbiota during infancy. During infancy, whether the gut microbiota will be reduced or they will be increased, that depends upon various multiple factors. The microbiota offers many benefits to the host. What are they? Through a range of physiological functions such as strengthening the gut integrity. Most important is strengthening the gut integrity or shaping the intestinal epithelium. The intestinal epithelium and the gut integrity, it is maintained because of the microbiota, and they also harvesting energy, protecting against pathogenic materials, antigens, and regulating the host immunity. However, there is potential for these mechanisms to be disturbed as a result of an altered microbiota. When these microbiota are altered, our whole metabolism goes haywire, and this is called as dysbiosis. So, dysbiosis is the mother of various diseases. Increasingly sophisticated methods are required to know about all these complex ecosystems. So, microbiota has a unique role and so many functions to perform for the integrity of the gut intestine. The same thing has been shown here, that the gut integrity depends upon the microbiota, and the various functions of gut are shown here, and you can see these functions are totally physiological, starting from eating something in the mouth, mastication, and then after mastication, the salivation, and the whole process starts from the oral cavity itself. Then the deglutition, which is again a physiological process. After deglutition, then comes the role of the digestion. Before digestion, it is mixed with the acid, and not only acid, also mixed with various other substances which are present in the gut. So in mixing, churning, all these occurs in the gut, and these are all functions of the gut which are carried out. Then comes the absorption, and for that SGLT1 are very important for absorption of glucose, and other mechanisms are there. Most of the time the absorption is passive in nature. The uh, major bacteria, phyla, and genera predominating in the human gut are shown here. I told you in the beginning that firmicutes are 60 to 80%. Firmicutes are 60 to 80%. Firmicutes basically belong to Rhinococcus, Clostridium, Lactobacillus, Enterococcus. They are basically cocci, and they are the most important bacteria present in the gut. Then comes the bacteroidetes, 20 to 30%. They are basically bacteroids, and you can say that they belong basically to Prevotelo and the Xylanobacter. Then the minor amount is Actinobacteria, basically the Bifidobacterium. This Bifidobacterium has been used for treatment of the IBS and other GI symptoms as well. When commercially pre-probiotics are given, the Bifidobacterium is the main constituent of those pre-probiotics. Major Again, then after that, the uh, proteobacteria, they are less than 1%, they are very less, and basically the Escherichia and enterobacteria is they are present in the gut. This study has been done in so many years to find out the constitution of bacteria in the gut. Now I was talking about the dysbiosis. That is, this ecosystem of the gut bacteria is disturbed. Basically, you can divide them into two parts: the good bacteria and the bad bacteria. The good bacteria are basically symbiotic, or they have some function to perform, and their number is usually high. But sometimes the bad bacteria become more in quantity, or their function become important, and this is will be called as dysbiosis. In dysbiosis, either the bacterial load has gone down or the bacterial type has changed. So both type of dysbiosis can occur. We have enough evidence that the dysbiosis has a definite relationship with the disease, and it has been seen that various diseases like external infections, they are basically because of the diet or prebiotic, probiotic, antibiotic uses, the illnesses, lifestyle bimariya, and the living in environmental problems. They are lead affecting our health, and the basically the density, diversity, and activity of the gut bacteria is changing, and when it change, the homeostasis got disturbed and becomes dysbiosis. This dysbiosis will lead to various diseases, and if these bacteria are corrected, how they are corrected? Either by diet, or by prebiotic, probiotic, or by the antibiotic, or by the various other metabolic methods, then again the homeostasis is attained, and that leads to the health again. Other factors which can affect this dysbiosis or homeostasis is the internal host properties, like the age and the genetic and the stress level, the physiological processes, and the anatomical changes occurring, and physiology of the digestive tract. If the physiology of digestive tract is changing, then this can happen again. Then the basically uh, this dysbiosis is basic cause of most of diseases which are occurring. The diseases which have been found associated with microbiota in physiological condition and also in disease condition are described here. Various diseases like fibromyalgia, diabetes, the rheumatoid arthritis, the various other conditions like atherosclerosis and the anxiety, depression, the common obesity, IBS and IBD, gut problems, motility related changes, and the some type of cancers, they have been found associated with with the number of these microbes or the number of the gut microbiota. Their constitution and the functions are going to affect the various conditions and diseases as well. The examples are given here, examples how disease caused by bacteria has evolved. The disease of 150 years ago, cholera and the pneumonia, the scarlet fever, the diphtheria, and the whooping cough, the tuberculosis, and the smallpox, they are very old diseases, and modern day diseases like obesity, diabetes, diarrhea, the atherosclerosis, the colitis, Crohn's disease, and various related problems like autism and the asthma, the eczema, and the multiple sclerosis. They are the newer diseases, and they have also been found associated with the gut microbiota and dysbiosis. The diabetes has long been studied and found related with the gut microbiota, and diabetes and gut microbiota dysbiosis is a convincing link. Now we very well know that diet affects the gut microbiota, and the gut microbiota are changed in type 2 diabetes. Type 2 diabetes itself can change gut microbiota, and various factors like the physical activity, the anxiety, the other problems related with that, they are changing the gut microbiota synthesis, and finally they can lead to dysbiosis. So, gut microbiome balance controls the long term health. In long run, it is the gut microbiome uh, balance which is going to balance the health as well. So, that's what is said commonly. Then, what is your take on this point that the gut microbiota is host and host metabolism are correlated? Then it is said to very truly that in transition from infancy to adulthood, gut microbiota changes and interacts with host metabolism, leading to metabolic and cardiometabolic diseases. So this is one saying. I think most of us agree to this that it occurs, and this is the deciding factor. Most of us know. Therapeutic modulation of gut microbiota from cradle to grave. The therapeutic modulation keep on occurring throughout the life, and in various conditions the gut microbiota is keeps on changing, and it has the circadian rhythm as well, that the gut microbiota will not remain same throughout the day. That is why it is said that a particular time one should take meal, and one should not take meal during night time, because the gut microbiota will not remain same and going to alter every metabolic process, whether it is controlled by other factors or not. It is immaterial. The multiple cited impact of gut microbiota on the host metabolism. This you can see here, the fat storage, the energy metabolism, and almost every mechanism is being affected. So the adipose fat, the skeletal muscle, the liver, the heart, the oral cavity, the brain, and the gut. Everywhere the gut microbiota can work, and they keep on affecting the dysbiosis, and that will lead to disease condition. The here the you can see the insulin resistance. More work has been done regarding insulin, regarding the diabetes, and you can see here the link between the gut microbiota and metabolism has been shown here, that the lean gut microbiota, or the gut microbiota which does not allow weight to gain, and this can lead to the various intermediate products, like the mechanism that will lead to changes at the fatty acid oxidation and by AMPK pathway, and the this will decrease the inflammation, and the short chain fatty acids will have the impact on lipogenesis as well. Then the incretin and secretin hormone secretion will be increased, and the here the Other mechanism can be the butyrate production will rise, and when butyrate production will be more, then the gut dysbiosis will not occur. In other words, in other side, the muscle and the adipose tissue, liver and epithelium, and large intestine are going to impact in negative direction, and all these mechanisms will be rising, and they will lead to dysbiosis, and then person will have the more weight, and again it is AMPK activity is decreased, the LPS inflammation has increased, and the SCFA has gone has increased, and the lipogenesis is rising, and it will also have impact that incretins and the incretin and the secretin will go incretin secretion will go down, and the butyrate production will go down, and this will make a person obese, while the other mechanisms which I taught in beginning are involved in making the lean person. The microbes and the host metabolism. The microbes may influence host metabolism, and this will can occur in various ways. One is the lipopolysaccharidases and the short chain fatty acids and bile acids. Three mechanisms are known. The lipopolysaccharides originates from the outer membrane of the gram negative bacteria and brings the to tall like substances. I think you all know it very well that what are the tall like substances, and these tall like substances will definitely alter the mechanisms involved in that. The This will activate the pro-inflammatory and inflammatory pathways, resulting to the low-grade inflammation and thus decreased insulin activity. Short chain fatty acids are responsible there, and these short chain fatty acids have been found in relation to in rise in the all these conditions, which can lead to dysbiosis. Bile acids, primary bile acids are produced by the liver, and the their role again becomes very important because they are going to increase the conditions where the these are required. Gut microbiota has also been talked about in relation with the cardiovascular health and disease condition, and it is said that cardiovascular health again determined by gut microbiota and possible molecular pathway is linked to cardiovascular and cardiometabolic diseases as well. Lately, this has been also used as a pharmacological agent to treat certain cardiovascular diseases. So, gut connections to the diabetes are well known, and it is a lot of studies are available where we found that the gut microbiota dysbiosis can lead to diabetes and difficult to control diabetes. Gut microbiota and obesity have a lot of evidences that the obesity is related to gut microbiota. Various mechanisms have been put forth, which are written here. You can very well see it is not only one organ, ranging from brain, liver, adipose tissue, muscle, and epithelium, all contribute for obesity. The gut microbiota and diabetes. This is well known, and a lot of research is available on this point. The high fat diet modulates the composition of gut microbiota, promoting the metabolic endotoxins, and these endotoxins trigger into this development of metabolic disorders. So that is very general mechanism talk about. Endothelium or endothelium toxicity or endothelin toxemia is associated with the increased risk of incident of diabetes that has been shown by various studies, including this one. The gut microbiota as a target for the treatment of type 2 diabetes has also been tried, and few trials are available that if given in good quantity, these pro and prebiotics can alter the glycemic status as well. Probiotics in metabolic syndrome have been tried. In beginning, I said that lot of preparations are available, and now we know that the gut microbiota can be changed by that, and they can also alter these conditions very well. Probiotics via the butyrate induce GLP-1 hormone secretion. So GLP-1 hormone secretion also alters by the gut microbiota changes, and this should be kept in mind. Patient with metabolic disorders like obesity, diabetes, a cardiovascular risk factor, they all should be the they can be called as immunocompromised patients. And here again the role of these agents will definitely more. Effect of multidisciplinary or multi-species probiotics supplementation has been tried for the metabolic profiles of the patient, and the multi specialty species probiotics of different species supplementation on metabolic profiles has been seen, studied as HSCRP oxidative stress in type 2 diabetes mellitus has been studied. And there is ambiguity in the uses of single strain and multiple strain probiotics and what should be done. The problem is that all these probiotics may not reach to the site where they are required. It can be said, the article was published in Times of India that said that the days is not far when doctors do gut profiling, give food plans to patients, saying that the this can cure your disease. This is said by the Stanford professor that this can happen in future. So that was from my side. Now I await for your questions, which can be in plenty. I will like to address them all.
Thank you so much, sir, for this informative talk. The PPT was so much contentful and informative. Sir, we have received few questions from our participating doctors. With all your
Yeah,
permission, can I put them across?
yeah.
Okay. The first question we have received, that is, how do the various segments of the gastrointestinal tract contribute to the digestion and absorption of nutrients?
Yes, the digestion of various nutrients starts from the oral cavity itself. When we take food, then in oral cavity it is mixed with saliva. That is why we advise that one should take food very slowly, and when the mixing of saliva occurs, then food becomes soft, and there are enzymes like salivary amylase, and these enzymes, the enzyme start working on the food, and the food digestion starts in oral cavity. Then it continues even in the stomach, but the exact digestion after stomach occurs in the intestine, small intestine, and duodenum. After intestine, the absorption also starts. So, for digestion, oral cavity, stomach, intestine, small intestine, and the pH of the various organs is different. So the different type of digestive juices are there. In GI tract, stomach has got more acid, and the enzymes which are working in acidic medium, they will be working in the stomach, and those enzymes which require alkaline medium, they will have their impact when the food goes into intestine. So in various phases digestion occurs. That is why good digestion involves the gut motility as well, and the good amount of enzymes and good pH, supportive pH for digestion. And for absorption, again, the small intestine is enough for absorption of various constituents. Duodenum is basically for the uh, absorption of various metals and very important minerals as well, while the large intestine, basically the water contents are absorbed in the large intestine.
Moving to the next question, that is, what are the mechanisms underlying gastric acid secretion and how do they impact the development of GI disorders like GERD and or peptic ulcers?
This is very important question. Peptic ulcer and gastric ulcer, nowadays they are rare, and a lot of PPI are being used. So various GI disorders. At present, one line I'm going to say that is very, very important, but some of doctors may not agree. That is, at present, PPI is not curing GI disorders, it is creating GI disorders, because acid suppression is not required all the time. Acid is very much required for the digestion absorption of various substances. So GERD is not only because of high acid, it is because of the wrong food habits. So without altering food habits, a lot of PPI and lot of prokinetics are being advised. That is not correct. So I will like to say here that the GI tract acid is not responsible for most of disease. It is responsible for development of peptic ulcer. But again, other factors of peptic ulcer are more important. Wrong food habits, wrong sleep habits, and wrong physical activity, and selection of food, and lot of mental stress. They are more important rather than acid secretion alone.
The next question is, what is the significance of the mucosal barrier in the GI tract and how does it protect against pathogens and toxins?
This is very, very important question, I will say, and I congratulate for this question. Basically, these mucosal barrier are very, very important for integrity of intestine and whole GI tract. And the mucosal barrier is strengthened by the gut microbiota, and that is the basic purpose they are serving in GI tract. And the mucosal barrier is very important. Unless mucosal barrier is there, the person cannot live.
Okay, moving to the next question. What is the relationship between gut microbiome and the development of colorectal cancer?
The direct relationship has not been studied, but indirect evidences are there that the colorectal cancer has some relation with food habits and gut microbiota as well. But regarding any cancer, single factor is not responsible. It is the collection of various factors, and food habits and the gut microbiota are the one of the important factor which decide the colorectal cancer.
Okay, we are taking the last question for today's session. How does the gut brain axis function and what is the role of the gut microbiome in this communication pathway?
Actually, gut also, it is also said that the gut has small brains. This is called the little brain concept. All the neurotransmitters which are present in the brain, they're also found in the GI tract and vice versa. The neurotransmitters which are present in GI tract, they are secreted by various glands. They are also present in the brain. So ultimately, the gut affect brain and brain affect gut. That is called as gut-brain axis. And this gut-brain axis is determined by the presence of good and bad bacteria inside the gut. So the gut microbiota will alter the stage of gut, and that will have the impact on the brain as well. That is why the probiotics and prebiotics are used to treat a psychosomatic disorder like irritable bowel syndrome.
Thank you so much, sir, for your expert opinion that you have shared. And I'd also like to thank all the participating doctors for your continuous participation at our platform. Thank you once again, sir, for your valuable time and for being with us at our platform and sharing such valuable insights with our doctors. With all your permission, can we just end the live here?
Yeah, yeah. Thank you.
Thank you, sir. Thank you so
Thank
much.
you.